Learning objectives include how cytotoxicity type 2 hypersensitivity occurs, and how to recognize it or diagnose it, and how to treat it. Well, type 2 hypersensitivity is also known as cytotoxic hypersensitivity. It involves IgG and IgM. Like type 1 hypersensitivity, we know involved IgE, but here two other antibodies are involved, either IgG or IgM, both, or both could be in the same case. Along with complement, this combination is very important. So there are antigenic cells basically in the body that against those antigenic cells, antibodies are produced by the body, and then those antibodies attach to these antigenic cells in the body, and then complement is activated. And once the complement is activated, it punches holes into the cells and damages the cells. So basically, in this hypersensitivity, the cells are damaged. And a very good example is blood transfusion reactions. If the blood is mismatched for some reason, or is not matched properly, the patient, when receives blood transfusion from another person, all the cells would die. They would be lysed because of these antigenic antibody and antigen response along with the complement. And as you can see here, RBCs have antigens on them. And it is very important that blood grouping has to be performed and the blood has to be matched before a transfusion can take place. This is an example. In this slide, you're looking at platelets as an example, as a model, that some antigen, that could be a drug, could be anything that you have eaten, mostly drugs, they sometimes attach to the surface of the platelets. This is just one example. These chemicals can attach to any cell, like RBCs, like platelets, like lymphocytes, like any other cells. And once these antigens attach to the cell, body recognizes these antigens as foreign, and body would start making antibodies against them because these are foreign antigens to the body. And when antibodies are formed in the body, these antibodies would attach then to the cells that have the antigen attached on them, on the surface. And IgG and IgM, it is the property of these two antibodies that they can activate complement. So the whole situation is a set for the damaging property of complement. So as a result, when antigen-antibody interaction takes place involving IgG and IgM, complement is activated. And the end result of this activation of this complement is holes in the cell. So the cell membrane is damaged, leaking the contents of the cell and causing the death of the cell. How to diagnose? As you can see, if this is a RBCs or these are platelets, for example, you can measure the number of these cells in the body. If there is an immune reaction or hypersensitivity reaction type 2 occurring against, let's say, RBCs or against platelets, for example, you can measure them, you can count them in a hemocytometer, and you would know that they are being decreased in the individual, in a patient. So that leads to a kind of doubt or kind of lead that this guy or this person is suffering from some immune-mediated deficiencies where immune system is working against the cells because those antigens are absorbed onto the surface and antibodies are trying to clear them. So low number of cells can give you a kind of clue, although it's not a confirmatory test until you do some other testing. But this essentially can lead to a point where you can confirm them later. I mean, this is a specialized field of immunology, but overall, you can get the hang of it. Thrombocytopenia is the result of when you lose your platelets. What is the treatment? Of course, if you know that this is the antigen, it's very hard sometimes in diagnosing this type of hypersensitivity, very hard, because looking at the antigen is really hard. So most of these patients, they are only treated symptomatically by giving some corticosteroids that decrease the inflammatory process. That is what we can maximum do it. If we know the antigen, then avoid the antigen. That is the treatment. So in summary, cytotoxic hypersensitivity involves either IgG or IgM or both. And there is a complement activation during this process that leads to the damage. And the patient could be treated symptomatically. Thank you.